Good afternoon, everyone, and welcome to Genetic Alliance's webinar series. Today we have a session, the National Center for Biotechnology Information, presenting Submitting Information about Variation to Public Databases. My name is James O'Leary. I'm the Chief Innovation Officer with Genetic Alliance, and I'm very pleased to have with us two staff scientists from NIH, specifically at the National Library of Medicine, NCBI. Today's webinar will talk about the benefits gained from submitting information about rare variation, what types of information are being requested, and current methods that are available to make submissions of all of NCBI's variation databases and websites. As a reminder, participants on today's call are muted. Questions and comments may be submitted at any time during the presentation, and we welcome you to do it throughout the presentation as you think of them. On the right-hand side of your screen in the panel. Today's presentation materials can be downloaded at geneticalliance.org/webinars, and today's session will be recorded, so you can view that webinar recording streaming live or downloading it off of our website probably about mid-next week. Also, as a reminder, please fill out the automated survey after you exit the webinar. That feedback really helps us and our speakers to make future webinars a success. So with that, we're going to turn things over to our speakers for today, who will go through their presentations, and then we'll have time for questions and answer at the end of the presentation. So thank you very much, Tim and Donna, and welcome.
Hello, everyone. I'm Donna Maglott, and I'm going to start the presentation today. And so I would like to add my welcome to this webinar. This is my first time, so bear with me because I feel like I usually require feedback from the audience, and so talking to this screen is an interesting experience. So let me go forward. What we're going to cover today, I've outlined here. One of the major topics is to go over why submit to centralized databases. We certainly feel that information that different groups may have about rare variation is not likely to be in some of the centralized databases now, because most of the research focus has been in gathering information about common variation. And so we certainly want to make it possible for interested parties to submit data about rare variants and what the phenotypes are that may be associated with those variations. Having those data centralized makes it universally accessible and can provide a good foundation for additional research or identification of variants to be tested, or the interpretation of a variation should someone be identified to have a particular variant. We'll also briefly cover what data we are processing for being in scope for centralized databases, what data can be provided without identifying individual patients, and finally the mechanics of how to submit data to centralized databases. So what I'm going to start on first is showing you some screenshots about what will happen when, if you contribute information that about rare variants to centralized databases, in the mode of showing you screenshots from how we currently report those data on the web. And so we're using as an example the ABCC6 gene because Tim is the database moderator for the Locus-specific database for this gene, and so we have a lot of information for this gene, and this is sort of an example of the benefits of submitting data. So what I'm showing you here is a screenshot of an interface that we call our graphic display of our sequence. And so if you look at this diagram, you can see that it's labeled with the gene symbol, that it's a sequence that's called the RefSeq gene, as I've boxed up on the top, and that there's a diagram that's relatively complex. Don't worry about looking at it now. I'll go into more detail later. But the major take-home message from this screenshot is to show that we can integrate information about all the submissions in a sequence context to make it easier for people who are doing research as well as doing testing to identify what might be known about variation on a particular gene. I'd first like to take an aside to talk a little bit about what a RefSeq gene is, because NCBI does create a sequence standard that we call a RefSeq gene. The URL at the top is for more information about the RefSeq gene project. And in essence, what a RefSeq gene is a standard gene-specific sequence in genomic sequence coordinates so that it can be used as a standard of reporting when an individual is tested and identified to have a sequence different from the standard. It also is a stable sequence so that as the sequence of the genome may change, we can have a standard way of representing where a variation is against a reference standard. These sequences are very important, and so to the greatest extent possible, when we generate a RefSeq gene sequence, we interact with gene-specific or disease-specific stakeholders to identify what sequence they may have already used as the standard and establish the RefSeq gene sequence accordingly. At present, the RefSeq gene project has about 3,600 records, and they can be retrieved from our public databases by this URL at the bottom just by searching our nucleotide sequence databases by the word RefSeq gene. So, in other words, one way to integrate information about sequence variation is to make sure that we provide that report against the RefSeq gene standard. Now, going back to the screen I showed you before, I want to point out that you can focus in on a particular region, so I'm going to go through these screenshots emulating the idea that you had identified an exon-specific region on the sequence that you wanted to look at in more detail, and so this happens to be exon 24. So in going to the next slide, this is now showing that we are zoomed to that highlighted region. This again is that same RefSeq gene standard sequence, but now this portion on this region shows that you're focused on a smaller region of that sequence, and this is the portion that is being displayed below. There's still a lot of boxes here that I want to explain, so let me go over that in more detail. Again, as I pointed out, there's a little icon at the top to show you what portion of the sequence that you were looking at, and this boxed region over here shows you how many base pairs are being examined. There are also possibilities on this particular interface to click and see some more details. So in this particular instance, I've established what's known as a marker to represent a particular variant that is annotated. This track right here labeled SNP is a report from the dbSNP database, SNP meaning an abbreviation for a single nucleotide polymorphism. These variants are not just single nucleotide polymorphisms because some of these are very rare variants and therefore by definition wouldn't be called polymorphic. But these annotations are color-coded so that if they're red, they're a single nucleotide change. If they're blue like this one over here, it's an insertion deletion. And so if you generate a marker that you can label, you can see on that marker up at the top box the sequence context that you have highlighted, and when you look at sequence records, they're all given a label called an accession. So in that top box, there's the accession column that gives the RefSeq gene accession. That starts with an NG. And then there's our reference sequence mRNAs and proteins that are also given, and there are two conventions for numbering systems. One is starting at the first position of the sequence, which we've labeled as sequence start, and that's and an R in the column that's headed HGBS name. And the other convention is by the starting of the coding region in column 3, that's relative to the CDS start, and then the number is given according to that convention. HGBS, in case you're not familiar with that, is an organization that stands for the Human Genome Variation Society, and that has established a numbering system and a convention for representing nucleotide variants. And this particular report is just giving the location according to those numbering system conventions. I want to point out that down at the bottom, what this is pointing out is if you hover over one of these little boxes, it will expose more details about what is in that box. And so let me backtrack a second here to get rid of some of these labels. What I forgot to point out before is in this portion of the display, there's a track labeled SNP for SNP. There's another track labeled LSDB, or clinically associated variants. So what each of these boxes corresponds to is in this window of the sequence, there's a number indicating the number of variations in that region that were actually submitted from a locus-specific database or are thought to be clinically associated. The third track here, labeled cited variants, gives within that same window of the sequence how many variants that are being annotated here have been cited in PubMed, and therefore how many references we have to the variations in those regions of the genome. So when I'm getting back to this part that's more highly expanded, if you hover over the annotation of the variant, what will pop up is a box that provides you a little bit more information about what's being displayed and provides as well information to show you where you can look up more data. So in this box it says gene symbol. There's a link to ABCC6 that goes to NCBI's gene database so that you can read all about the gene. The position is reported. There's a report to the identifier that the database called dbSNP assigns to this particular variant. Each variant is given a standard identifier, so that is the identifier for this variant in the dbSNP database. And there is also a link to something that's called variation viewer, and it's giving the gene symbol that is being reported. The point in all of this is that having the sequence data and knowing where the variation is, we can have the positions or the reports of particular sequence variants, which I'm calling sequence annotation down here at the title of the screen, can be used as an organizing principle for displaying information about sequence variation and where you can find out more about the variants that have been represented on the sequence. This is a screenshot from the variation viewer, which can be accessed from the sequence display that we were looking at previously. The variation viewer has a name at the top for which gene is being displayed. There's a table that you can page through that gives information about each variant. And then there are other columns that provide links to more data and information about the submitter. And so this particular report can be focused on all the variants that are thought to have some clinical association, but you can also in this type of display report all variants that have been submitted on a particular gene. Going to this in a little bit more detail, the kind of information that is available from this variation viewer. You can get a report of the type of variation in that first column. You can get the report of the location of the variation in the HGBS system so that you can see what the sequence change is that's being reported in all the different numbering systems. You also get a column of whether this has been reported to be pathogenic or not. And then there are more columns about how many times this has been reported and the number of observations. And if there's a link here, you can get information about the allele frequency. And then there's a whole set of links to additional information, whether it be in the published literature in PubMed, in OMIM in the allelic variant section, so that you can see links to OMIM here, or to dbSNP based on the RS number. And finally, this last little icon gives you a link back to the sequence viewer, in this case from whence we came. The final section here indicates who deposited information about this variation, and then sometimes there's a link back to the submitter resource so that you can look up more data. So we are certainly keeping track of who submits the data and provide links back to those resources as well. If you follow the link to OMIM on the allelic variant, you can read more about the description of each allelic variant as has been described by OMIM. This is a screenshot from a section of the allelic variant part of an OMIM report. And you'll notice that since NCBI also distributes OMIM, we can add in links that OMIM may not have already reported. And so when we have made the connection between a record in OMIM for the allelic variant and a record in dbSNP, we provide that link back to dbSNP. So again, if the data are submitted and we attach some these descriptions to the genome and all the cross-referencing data, then if a user comes to NCBI from the point of view of looking at OMIM, they can be directed to dbSNP and to the sequence data. If you follow the link to dbSNP, if you've ever been to dbSNP, you know the records are very complex. I'm not going into all the details here, but the major take-home message is if there is a variant that is reported to be clinically associated, we indicate at the top. We provide links back to the variation viewer, as I was already summarizing, or back to OMIM. So no matter where you start in looking at all the data, we provide navigation to additional information. So again, by having the data submitted with all the descriptive information that are possible, many users can find the information, whether they're a researcher and looking at things from the dbSNP point of view, whether they're looking at OMIM from the clinical side, and so forth. And so this pop-up is just summarizing that as well. In dbSNP, you will get information about and links to more information. If you scroll down on any record in dbSNP, you also will see that the submitter data, the sequence, and links back to OMIM. So what I've tried to do in all of those screenshots is demonstrate if you submit data to centralized databases, that is integrated into all the reports that the centralized databases provide, and no matter where people might be accessing those centralized resources, they can find information that you submit. The next point that I wanted to touch on briefly is what kind of data are in scope. I think the display that I provided for the variation viewer summarizes that most explicitly. For any sequence change, we want to know how that sequence change might affect health, how often that change has been observed, where you might find more information, and certainly give credit to the submitters and to the submitters resource for more information about the submission. Now that we've tried to summarize what the benefit, how access can be provided, let me show you a brief summary of how you can submit data to centralized databases. In essence, we'll be talking about two major mechanisms. We do have some web-based interfaces to submit data, and then Tim will be talking more about the mechanism of creating a Locus-specific database and submitting from that Locus-specific database. This slide just summarizes the major web interfaces that we have for submitting data. If you just know one variation and you want to submit it, we do have the web form that's provided at this URL. There's information there about how to make the submission. The critical aspect is you need to be able to know what your variation is according to a known sequence so that we can connect it to the sequence data, and the information about how to do that is on that website. If you have a record of a lot of variations and you want to fill out a spreadsheet on these, we do have a mechanism that you can upload data via an Excel spreadsheet or a tab delimited file. And these URLs are pointing out where the submission form is, and that there is documentation on our bookshelf on how to submit those data. RefSeq-Gene is part of a collaboration with a group at the European Bioinformatics Institute, and the sequences that we're establishing as standards are also part of this collaboration with the acronym of LRG for Locus Reference Genomic. I provide here the URL for that website, and that resource also provides guidelines on how to submit, but they eventually come back to NCBI and we process them here. And as I said, Jim will be talking more about how to submit via a Locus-specific database. I point out that there's an email contact information down here for if you have questions about submission, questions about RefSeq-Gene sequences, and so I encourage you to take advantage of that email. So finally, I want to point out that submissions to NCBI are processed in large part through these steps, that you as the submitter will organize the data for submission, enter the data in a spreadsheet or on the web form. When we receive those data, we will validate them. In other words, make sure that we can identify what sequence that you're talking about. If we can validate those data, we will assign identifiers to them. If we have trouble with it, we will get back to you and ask questions so that we can make sure that your submission is processed as you want it to be. Then, depending upon what you submit and how you want it to be processed, we will release the information. It will be available on our websites, and we also make it available for automatic transfer via FTP. And one of the benefits of submitting to a centralized resource is when we make these data available for FTP, the data are picked up by major other database systems such as EBI and the genome browser at Santa Cruz. And so once they are submitted, all browsers will end up being able to have information that you have submitted. So now I'm going to segue to Tim, and he's going to give the rest of the talk.
Hello, everybody. My goal today is to tell you about the LOVD, or the Leiden Open Variation Database. This is a database in a box software package that is intended to be a streamlined way for you to load and represent the variation for your disease gene of interest. And to just tell you about our experiences of doing this with the ABCC6 gene mutated in a connective tissue disorder, rare disorder called pseudoxanthoma elasticum. And I just hope to give you an idea of what our experiences have been and what you can expect if you choose to use this as a way to submit your data. Just to give you a few details about ABCC6, gene was discovered in 2000. It's autosomal recessive disease, carrier frequency of about 1 in 150. The gene is expressed in the liver and kidney, and there are 2 partial pseudogenes that we have some experience trying to relate that variation as it pertains to clinical variation due to mutations in the ABCC6 gene. Just to give you an idea of the scope of this particular project, there have been 239 mutations found to date, and as you can see, the distribution of these mutations. And if you include non-clinically relevant variation, synonymous mutations, which may or may not be clinically relevant, but also intronic variants, then the total variation to date is 378 mutations. And if you are like we were, then you probably have all of these in a spreadsheet somewhere in some format that you devised yourself, and you may be looking for a way to present this to the public in a locus-specific mutation database. So some things that you might want in a database, that it be open to the public, and that it be stable, that it have accessions that are stable over time so that you can things aren't changing on you all the time. It should be a comprehensive catalog of all of the variation for your disease gene of interest. Should all be in one place. Should be user-friendly, searchable, customizable, and ideally it should be free. And the Leiden Open Variation Database, LOVD, is all of these things. Here is the home page of the ABCC6 database, hosted at NCBI. We are experimenting with this as a way of communicating and representing disease gene variation. There, in fact, are a great many more examples of this at the LOVD home itself in the Netherlands. But this is just to give you an idea of what our homepage looks like at NCBI. You have some basic information about the gene, its location, and so forth. You have sections on all of the variants that you can see. You can search the database, and there are external links to reference sequences and additional tools and resources that you can turn to find out more information about this disease gene. You can also access each of these through these tabs. I don't know how well you can see them at the top. Variants, who may have submitted mutations for this gene. You can submit yourself, and you can read the documentation about the LOVD package, software package. And this is just, if you want to take a look, you go to the main NCBI page /LOVD. And this is what a variant table looks like. It's just an example. There are any number of additional columns that are optional. This is just to show you the mutations themselves, then the fact that you can represent them in HGVS nomenclature as Donna described. You can sort by any one of these columns the type of mutation that it is, by the protein mutation name, exon, intron, etc. We also, and you have a link to the article on PubMed that was originally described the mutation when it was first found. Additionally, you have unique database identifiers for each of these mutations. And in the case of ABCC6, we still have yet to load this, but we can annotate pseudogene variation. And once dbSNP accessions are assigned, then this table will be completely populated. There is a link, of course, to submit any new new mutations. You just provide as much information as you have. Right now, we don't have, or LOVD does not have a way for you to submit the raw data, such as sequence traces and so forth, but we do encourage you to also submit that just so that, as a submitter, I should say, so that curators can confirm the authenticity of any variants that you're reporting. And once your data submissions are reviewed by curators, then they would go to the public LSDB site. And again, just to reiterate that you may be familiar with seeing mutations in the form of something like R1141X, whereas HGVS nomenclature provides an unambiguous nucleotide resolution detail of what this mutation is called. And there's a plug-in module within the LOVD software called Mutilizer that will either generate this information for you or confirm it if you have not already put it in this format. Just some caveats about working with LOVD, again, based on our experiences. If you intend, as a curator, if you intend to make multiple changes at once, as you can do, for example, in a spreadsheet, this can be challenging. The LOVD is suited to making sort of single changes at a time, but if you want to make multiple changes, for example, fill in a whole new column with data on all of the variants that already exist in the database, you do have to download the entire database, make your changes in a spreadsheet, which often has formatting issues. You have to empty the database and then re-upload the entire database from the spreadsheet. So it takes some getting used to, but it certainly can be done. For those reasons, you need to have sort of long-term committed curators, people who are going to commit to doing this over the long term, because as new submissions come in, of course, you're going to want to have them put in to be included with all of the existing information. Another caveat is that we are experimenting with is the amount of flexibility involved with any clinical information that you may want to include with your data. There are certainly clinical fields. The way that these are typically entered is as a patient record is entered, and you enter which mutations this patient has, and then sort of, in most cases, because this is intended to be open to many different diseases and many different variations, the clinical fields that exist do tend to be fairly generic. And you can add your own, but that gets complicated when, for example, at NCBI, we have a single installation. If you want to add clinical information that's very specific to your disease, that gets added to the optional columns of anybody else who also has an LOVD database at NCBI. So we're just experimenting with figuring out the best way to express the clinical information. all of your variants, if you choose to go the LOVD route to submit your variation, will eventually end up in DBSNP and DBVAR. If you're not familiar with it, DBVAR is the new database of genomic structural variation at NCBI. It's sort of the counterpart to the single nucleotide variation database of DBSNP. And finally, I just wanted to say that LOVD is a good way to give microattribution. This is an initiative being put forth, as you can see in this article by at Nature Genetics, a way of providing an incentive to people in the research community in the form of immediate recognition of the contribution that they may be making with disease gene variants. And it's hoped that this sort of community annotation will result in a very high quality record of all of the different disease-associated variation. And this can only really be accomplished by people who care about the disease genes that they work on. And so this provides a method an incentive, rather, to come up with a very high-quality genome annotation. And I think with that, we'll turn to questions. If the moderators can relate your questions to us, then hopefully we can give you some answers. Thank you.
Great. Thank you so much, Donna and Tim. Just as a reminder, please submit your questions on the right-hand panel, and then we'll relay those questions to the presenters. So first off, a more general question that someone asked, which is, what does a committed curator look like? So I'm assuming they're asking, what would that individual look like in expertise? Where would you find such an individual if you were interested in developing this type of curated LOVD database?
Well, I think it would be somebody who has a continuing presence in the research community of whatever disease we're speaking about, and really it's somebody probably who it's not their primary job, and it's sort of a volunteer basis, but it's somebody who should have as I say, sort of a long-term involvement with the research community for a given disease gene. Because it does take, it takes some effort to maintain the data and to ensure its quality. I guess that's that's all I can say.
Great. Another question was, it says, it appears that several databases covering different types of variations are developing. Can you comment on that?
Could you be a little more specific, please?
So the person that submitted that question, if they can give a little bit more detail. While they can get back to us with a little bit more specificity, what alternatives are out there for LOVD? What are people using? What spurred you to develop this specific system?
I think we wanted to try this as a way of having this at NCBI, because NCBI has so many resources linked to disease gene variation, that is a way to have things here locally and to have it sort of intimately linked with a lot of other databases at NCBI. That being said, NCBI does not have the resources to host a lot of these locus-specific databases, again, because, as I indicated, it requires commitment from a curator, and we don't have these curators; they have to come from the community. And I think, as I also mentioned, the LOVD home in the Netherlands is host to, is it 1,000? Many. I'll just say many. Examples of LOVD. And so whatever system they've come up with for being able to host these and curate them, I'm less familiar with.
Well, I just add that part of the rationale for selecting LOVD as opposed to other interfaces for collecting the data. Is that it is open source. They were willing to work with us to make sure that the security of the website was consistent with our security standards, and they're working closely with a European initiative to manage genotype-phenotype information, and so they're being very active and very responsive to the community about maintaining gene-specific databases. So the major developer for LOVD, their group started out because they were themselves curators of locus-specific databases, and they knew what kinds of information they wanted to capture, both from the point of view of the variation and the patients that might have these variants. And so they have continued to work on a project that would facilitate gathering the data and making the data generally accessible.
Okay, excellent. So a follow-up from that earlier question. So it appears that several databases covering different types of variations are developing. Is there an effort to homogenize the databases? And then as a follow-up, do any of the databases follow emerging data structures from groups like HL7 for clinical use of genomic information?
So we are. So if you're referring to the fact that there are databases of different names like DBVAR and DBSNP, yes, we are trying to make certain that we are integrating information. That's one of the benefits of taking the submitted variations and mapping them to a standard sequence, because then we can identify when different reports overlap and be able to identify, therefore, that we're talking about the same variant. We have also we are very aware of all the different requirements for reporting sequence variation. We have worked with some of the groups in the National Library of Medicine for identifying what the standard report should look like for reporting a particular variation. So that's certainly one of our objectives, to make certain that the information that is captured in our public databases is compatible with all reporting systems.
Okay, excellent. Going on to a slightly different question, is there a good way to batch convert CRO position coordinates to HGVS nomenclature format?
I'm sorry, could you repeat what position formats?
Chromosomal position coordinates, what they're asking?
Yeah. Yes, so that's one of the submission formats. So HGVS as a reporting system, when it's in genomic coordinates, I gave you the example of a RefSeq gene coordinate system, but we align those RefSeq gene coordinates to chromosomes. And so if a submission comes in on chromosome coordinates, if we can find the appropriate sequence, we can convert it to all the others.
Excellent. Another question, wouldn't it be useful to list if the variant is somatic or germline when providing a clinical interpretation in your variant viewer? Or is the viewer showing only germline variants?
We are in the process of exposing when we know that a variation is germline or somatic. It's one of the data elements that we capture. Unfortunately, we haven't been exposing it as robustly as we should have, but that is a high priority task, and we hope to get that published shortly.
Excellent. These are like the quick fire questions, right? The next question is, how strict are your criteria for determining pathogenicity? For example, sometimes a variant will be reported in the literature in a case, but it turns out that this is a common polymorphism in the population. That's obviously something that we're very concerned about, that we are relying on submitter, and this is one of the benefits of having curation, that it's a domain expert for saying that something is pathogenic or not. The point is well taken that a single report in a single paper that a patient may have had this variant doesn't mean it's pathogenic,
And we are hoping by aggregating information from multiple submissions that we're going to be able to identify those that are truly benign versus those that are truly pathogenic. Again, one of the benefits of submitting the data centrally on what might be identified as being rare variants, if some other large-scale projects identify that this is a common polymorphism in people that are do not seem to have any overt phenotype, then we can discount. We have the foundation then to say that although this has been reported by pathogenic, there are many more reports to the contrary. So the only way we can do this is to be able to aggregate the information that might come from different data streams and look for consistencies and inconsistencies.
Right, and in terms of the LOVD database, that's all the more reason to find a curator who is, as I say, intimately involved and cares about the disease gene of interest, because ultimately they may be the ones who are responsible for ensuring the quality of any pathogenicity calls. And in the case of ABCC6, I can tell you that great care has been taken to be very conservative and very careful about looking into and reporting whether the variants are pathogenic or not.
Okay, excellent. Thank you. Another more general question that we received asks, what are other barriers other than the lack of available tools and curators to the submission of rare variants from rare conditions? So you had talked. So I assume this is referring to the fact that you had talked about that capturing these variations have been more difficult. You talked a little bit about the benefits of submitting that type of information. What are other barriers that exist that maybe aren't overcome by this type of database, but the community, I guess, could take some sort of forward motion or collective movement to help you get this information?
So I would say one barrier perhaps is identifying unambiguously what has been reported previously. By that I mean that if there's a report in the literature about a particular variation and it may be giving an amino acid change, it may be a missense mutation or something like that, the numbering system for the protein that existed at the time of that particular paper may not be according to current standards, and so that's when I was alluding to the fact that when we receive a submission, we then validate it to try to make certain that we can identify that sequence at the position in which it is reported. So I would say that one barrier for us for the limited work that we do in trying to pull out stuff from the literature or other resources to get things submitted into current sequence coordinates is indeed identifying what numbering system was being used and what the reference standard was, because that's not usually something that is required to be included in the published literature. So that's where the benefit of having a dedicated curator or someone who's really knowledgeable of something may help, because they might know a particular variant by a trivial name in which they happen to know that the numbering system was based on something completely different from the current HGBS standard and will be able to transform it more rapidly. For the generalist database like NCBI or any of the other major aggregating databases that's harder for us to do, because we might get a submission, no, we can't find it on the genome and can't process it, so we have to turn it back.
And I would also say, from the perspective of locus-specific mutation databases, is coordinating with other groups who may be collecting data on the same gene you are. So there may be, multiple different groups, some in Europe and some in the U.S. and some elsewhere, who may be sort of priding themselves as the database. And if you're going to curate and maintain something like the LOVD database, then you really have to get together, I think, and determine, who's going to be the curator, or what team of curators are you going to have, and find a way to consolidate all of your data so that there are not multiple instances of variation data on the same gene.
Okay, excellent. So I have an additional question, and if anyone else has more questions, please submit them using the panel on the right-hand side. My question is about the types of groups or researchers that approach you to use LOVD. Is it researchers associated with disease-specific groups? Is it academic labs? Who are the primary users of this tool?
Well, as far as the NCBI experience, we've just worked with PXE International to start with ABCC6. Donna, do you know about the other few groups who have?
So the other groups that have contacted us have been groups at NIH. So we've offered and worked with a small number of groups, but it's not as if a lot of people have approached us to do this.
Okay, great. But any of those types of groups are the types of groups you were looking to work with within NIH, outside groups and researchers, disease-specific groups, any of those types?
I would say all of the above.
Great.
And again, the LOVD in the Netherlands has many installations, or rather many different databases, and perhaps a quick email to their helpline could tell you more about what kinds of groups approach them for hosting these databases.
Yes, I would say, just to expand on that slightly, is in that mode of having a limited number of locus-specific databases for the same gene, the group at Leiden has established the framework for a lot of locus-specific databases, and they are looking for curators. So there is another way of approach submitting is to see if there is already an infrastructure set up for a gene of interest and register yourself as a submitter or whatever, and work with that group to generate the locus-specific database, because as part of the LRG-RefSeq gene collaboration, we are working with the LOVD software group in general to facilitate submission from the locus-specific databases into dbSNP and dbVAR.
Okay, and you had mentioned, so the group at Leiden, you had mentioned the Human Genome Variation Society. Are there any other groups active in this area that people should look out for, or if they're interested, look into joining?
Genetic Alliance. Although I guess everybody here already knows about that.
And we didn't pay you to say that, right?
No, I was off the top of my head.
Great. Okay. Go ahead.
I was just going to say there are a lot of overlapping interests. So certainly the Human Genome Variation Society tries to keep a list of locus-specific databases and who their curators are and what the paths are to the databases that are provided. I mentioned that the European group is organizing a resource called Gen2Fen, and they are also coming up with a directory of locus-specific databases in collaboration with HGVS. And at NCBI, in the Entree Gene resource, we do keep track of if we know if there's a locus-specific database, because we had to look up all of this in trying to identify the stakeholders for the RefSeq gene product. I would say that there are many more RefSeq genes than there are locus-specific databases, so there's a large number of genes in which there is at least some interest from the point of view of testing and or variations that have been reported to cause disease, and for which there's not a locus-specific database yet as far as we know, but we are in tune with all of these communities to try to make sure that we are not duplicating effort, that we're cross-referencing each other appropriately, and are integrating information as much as possible.
Excellent. And I think there are some other groups like Human Variome projects that put out bulletins and different information around these projects as well.
Yes.
At this point, it looks like, let me just, it It looks like we don't have any further questions. Tim or Donna, are there any final thoughts you'd like to share?
No, I just think I would encourage people, if you're thinking about, if you have access to a wealth of data on rare variation, then get it out there through one of these mechanisms, certainly through NCBI's variation viewer or an LOVD database. Get it to where people can use it and see it.
And as well, if you are looking at some of the data that are being reported publicly and you find that it looks confusing to you or that there needs to be an improvement, don't hesitate to contact us, because we certainly want to make sure that the data that are being provided are accurate and up to date.
Yes, and that is a big issue. We experienced with the ABCC6 database through careful curation of the literature that about 25% of the submitted data were actually incorrect, and we had to go back to the submitters and ask them to correct them. So, be very careful in curation of your data.
Excellent. Well, I really want to thank all of our participants today and those who submitted questions, and very much especially Tim Hefferon and Donna Maglott for joining us, for answering all of our questions. If you do have any further questions for them, I know they put their information on their slides, which are available online, or you can always email us at Genetic Alliance, and we'd be happy to connect to you. As a reminder, don't forget to fill out your evaluation, and these presentations, as well as those from previous weeks, are available on geneticalliance.org/webinars. This one will be up in about a week and a half. So thank you so much to everyone, and we look forward to hearing from you on future webinars.
Thank you.
Thanks. Goodbye.
Bye.